Hello students, how are you doing? Welcome to another biology class. Today we will learn about the function of mitochondria. Before we start, make sure you have subscribed to this channel. Alright then, let us begin. Function of mitochondria. The most prominent roles of mitochondria are to produce the energy currency of the cell, ATP, that is, phosphorylation of ADP, through respiration, and to regulate cellular metabolism. The central set of reactions involved in ATP production are collectively known as the citric acid cycle, or the Krebs cycle, and oxidative phosphorylation. However, the mitochondrion has many other functions in addition to the production of ATP. Energy conversion. A dominant role for the mitochondria is the production of ATP, as reflected by the large number of proteins in the inner membrane for this task. This is done by oxidizing the major products of glucose, pyruvate, and NAD, which are produced in the cytosol. This type of cellular respiration, known as aerobic respiration, is dependent on the presence of oxygen. When oxygen is limited, the glycolytic products will be metabolized by anaerobic fermentation, a process that is independent of the mitochondria.
The production of ATP from glucose and oxygen has an approximately 13 times higher yield during aerobic respiration compared to fermentation. Plant mitochondria can also produce a limited amount of ATP either by breaking the sugar produced during photosynthesis, or without oxygen by using the alternate substrate nitrite. ATP crosses out through the inner membrane with the help of a specific protein, and across the outer membrane via pores. After conversion of ATP to ADP by dephosphorylation that releases energy, ADP returns via the same route. Pyruvate in the citric acid cycle. Citric acid cycle pyruvate molecules produced by glycolysis are actively transported across the inner mitochondrial membrane, and into the matrix, where they can either be oxidized and combined with coenzyme to form CO2, acetyl-CoA, and NAD, or they can be carboxylated by pyruvate carboxylase to form oxaloacetate.
This latter reaction fills up the amount of oxaloacetate in the citric acid cycle and is therefore an anaplerotic reaction, increasing the cycle's capacity to metabolize acetyl-CoA when the tissue's energy needs, for example, in muscle, are suddenly increased by activity. In the citric acid cycle, all the intermediates, for example citrate, isochitrate, alpha-ketoglutarate, succinate, fumarate, malate, and oxaloacetate, are regenerated during each turn of the cycle. Adding more of any of these intermediates to the mitochondrion therefore means that the additional amount is retained within the cycle, increasing all the other intermediates as one is converted into the other. Hence, the addition of any one of them to the cycle has an anaplerotic effect, and its removal has a cataplerotic effect.
These anaplerotic and cataplerotic reactions will, during the course of the cycle, increase or decrease the amount of oxaloacetate available to combine with acetyl-CoA to form citric acid. This in turn increases or decreases the rate of ATP production by the mitochondrion, and thus the availability of ATP to the cell. Acetyl-CoA, on the other hand, derived from pyruvate oxidation, or from the beta-oxidation of fatty acids, is the only fuel to enter the citric acid cycle. With each turn of the cycle, one molecule of acetyl-CoA is consumed for every molecule of oxaloacetate present in the mitochondrial matrix, and is never regenerated. It is the oxidation of the acetate portion of acetyl-CoA that produces CO2 and water, with the energy thus released captured in the form of ATP. In the liver, the carboxylation of cytosolic pyruvate into intramitochondrial oxaloacetate is an early step in the gluconeogenic pathway, which converts lactate and deaminated alanine into glucose, under the influence of high levels of glucagon and norepinephrine in the blood. Here, the addition of oxaloacetate to the mitochondrion does not have a net anaplerotic effect, as another citric acid cycle intermediate, malate, is immediately removed from the mitochondrion to be converted to cytosolic oxaloacetate, and ultimately to glucose, in a process that is almost the reverse of glycolysis.
The enzymes of the citric acid cycle are located in the mitochondrial matrix, with the exception of succinate dehydrogenase, which is bound to the inner mitochondrial membrane as part of complex 2. The citric acid cycle oxidizes the acetyl-CoA to carbon dioxide, and, in the process, produces reduced cofactors, 3 molecules of NAD and 1 molecule of FADH2, that are a source of electrons for the electron transport chain, and a molecule of GTP, which is readily converted to an ATP.
That is all for today, folks. One last thing, before you go, make sure that you have subscribed to our YouTube channel and be kind to like and share this video. This video is edited by Biruk Solomon MSc in Genetics. I will see you in the next video. Goodbye!